All right, chapter 13 is going to give us an introduction into the cardiovascular system, but the cardiovascular system is going to traverse into the Unit 4 material. So there's a lot to talk about, but I wanted to include it here because it's primarily a muscle, and so I'm going to lump it in with all of our muscle components. Where we're headed between the third and the fourth unit is that the cardiovascular system is not just the heart. That's what we're going to study here in chapter 13. And you have an idea. The purpose of the heart is to set a what? What does it stand for? Pressure gradient. Okay? To pump blood. We're going to pump blood from which pressure to which? Higher to lower. Very good. Right, we're going to propel blood down a pressure gradient. Through these structures called blood vessels, which we'll study in chapter 14, in Unit 4, wrapping up the semester with. So all of our blood vessels are built kind of similar, but there's some profound differences, so we'll see how they contribute to the distribution of blood. And we can't forget a very important part of this cardiovascular system is what? What's the tissue, not an organ? Blood. Okay, we can't forget about blood. And we'll talk about blood extremely briefly. Chapter 15, really not giving it the credit that it's due, but I'll give you enough to kind of have an idea about the components of the blood. Okay, so here, chapter 13, we're going to look at the heart. Unit 4, we'll finish our discussion with the cardiovascular system. If you remember from lab yesterday and Monday, we learned that even though we have one heart, one set of blood vessels, right, one volume of blood, functionally we can look at the blood in two different manners. What were our two circuits? We had our pulmonary and our systemic. Very good. Pulmonary circuit serves the lungs, and the systemic circuit serves our body systems. Okay? And then even though we have one heart, we said we can functionally divide it into a left versus a right half as well. The right half distributes blood to which circuit? Pulmonary. So thusly it must receive blood from the systemic. And then reverse for the left side of the heart. What else? I was about to add something there, but I've forgotten already. Alright, so let's summarize for figure 13 too. And I'll make reference back to this image into the fourth unit because it's kind of a nice little simplistic view of the cardiovascular system. I think I know what I was going to ask. Entering which system is our blood pressure? So entering the systemic or entering the pulmonary circuit, would our pressure be the least? Entering the pulmonary, okay. And it'd be greatest exiting or entering the systemic circuit. Technically, the blood pressure is the least when it returns to the right atrium. It is the minimal amount of pressure as blood. So we have a high pressure, blood propels out and back right to the heart. We already summarized the difference between the pulmonary versus the systemic circuit. We learned that the pulmonary circuit gets its blood from the right side of the heart and distributes blood to the lungs. And the primary reason is to do what? Gas exchange. Including what? CO2. What are we doing with CO2 here? Getting rid of it. The word we'll use is unloading. We're unloading carbon dioxide and taking in, or you could say loading oxygen. Conversely, our systemic circuit, it does originate from the left ventricle, where our blood pressure is at the greatest, and takes the blood to the rest of the body to serve our tissues. What type of gas exchange happens at your systemic circuit? Primarily what? Releasing or unloading oxygen, very good. And loading CO2. So it's the gas exchange is reversed, which we'll pick up next week when we look in the cardiovascular system. Or excuse me, not the respiratory system will align with it. Okay. All right, so our pulmonary and our systemic circuit. Some terms that we're going to introduce now, but better defined in Unit 4, include pressure and resistance. Pressure is the physical force on the vessels of the vessel described here as walls, okay, or on the blood vessels themselves. What are we going to measure it in the body? What's our unit of measurement here in the pressure in the body? We use millimeters of mercury. Okay, so we're going to measure it in millimeters of mercury. And then we're going to generally talk about resistance. We're not going to quantitate it any. But what do you think the term resistance means? You ever felt any resistance in your life? What does it mean? Does it mean everything's good?
Opposition.
Okay, opposition, right? Opposition, I think that's a word. I use the word deter. Okay, so opposition. Okay. There is an opposition to blood flow in the body, which at first doesn't make sense, but we'll see that there's natural resistance or opposition factors in the body that exist just to get blood from point A to point B. So we're going to introduce the terms now. When we come back from the exam, after we take our exam on Monday, we'll better define pressure and resistance. But right now we'll say blood pressure, just force measured in millimeters of mercury, and resistance is any type of hindrance or opposition to the flow of blood. And if we were to really just kind of oversimplify the two systems, we'll learn that the pulmonary circuit actually has lower pressure when contrasted to the systemic circuit and lower resistance factors. So in other words, it doesn't take a whole heck of a lot of work to propel the blood to what? Where's it going to? It doesn't take a lot of work to pump the blood to the lungs, in contrast to the rest of the body, to the systemic circuit, where the pressure is extremely high, upwards of 120 millimeters of mercury, and the resistance factors are numerous. So high pressure, high resistance, and therefore the left side of the heart has to work what? A lot harder. Okay? And so what we're going to do in our anatomy look in lab today and tomorrow is we're going to see a profound difference in the musculature between the right ventricle and the left ventricle. Which ventricle should be needing less muscle to do the power? The right, and the left ventricle would need so much more. And you may be surprised at how thick that vessel, I guess a ventricle is what I need to say, is compared to the right side. What's the name of the vessels, or keep misspeaking today, what's the name of the chambers on top of the ventricles? The atria. The atria still have what type of muscle in them? They're also, that's our heart. So there's cardiac muscle. It's cardiac muscle, but does it need to, do you think it has a lot of resistance or need a lot of pressure to propel blood from point A to point B? No. And so the musculature of the atria, size-wise, tiny. Okay, really, really tiny. Okay, when you contrast, but it's still it's in the heart. It's cardiac, right, tissue, but it's just not as thick when compared to either ventricle because there's less pressure, less resistance. If we look at the structure of the heart, we've already learned from lab. There are 4 chambers. There are 2 atria that sit on top and 2 ventricles that are below that. There are also valves, which you guys identified in your pre-lab from Monday and Tuesday. The atrioventricular valves, we can abbreviate AV. You can call them AV valves. And then you also have the semilunar valves, which appear to be moon-shaped or crescent type of shape. Where are the atrioventricular valves located? They're located between the atria and the ventricles. And they're these really cool looking flap-like structures. When we get into lab, right, the tissue that we're looking at is from it's dead, right? It's preserved, and so it's not real. It looks kind of weird, right? And so when we look in there, you'll look for these things. What are those called? Chordae, tendonae. Okay, they're tendons that look like cords, right? Anybody else would call them the heartstrings. Okay, and what type of tissue are they? Connective tissue. And so when we get in there and we touch the heart, I want you to see if you can touch that and feel how kind of slick feeling it is, if you will, and then contrast that to the musculature of the heart. Okay. If we contrast the AV valves between the atrium and the ventricles, and then compare that to what's considered to be the semilunar, the bi versus the tricuspid valves, where are the semilunar valves located? Where? But you may have, I've got old lady ears. I need to try to get hearing aid. What did you say? They're called lunar because of their appearance. Where are they located? Did you say lumen? Where are they located? Between, at the arteries, atop the what? They're not atop the atria, actually. They're atop the ventricles. The semilunar valves are what controls blood between the ventricle and the pulmonary versus aorta. Pulmonary artery versus the aorta. The pulmonary trunk, right here. You remember learning that in anatomy? We'll be able to see the pulmonary trunk on our samples. The aorta, it's not as neat looking like this. In fact, our hearts that we're going to look at are going to be like squished in a bucket. You're going to like pull it, peak, and all kinds of stuff. So, and the aorta is usually where it exits right back here is kind of cut off, and it's kind of a mess. But in an ideal situation, you can see where the semilunar valves are located between the right ventricle and also the left ventricle. All right, so we look at the heart basic structure. We have 1, 2, 3, 4 chambers. We have 4 sets of valves. What do you think the job of a valve is to do? Keep the blood from mixing, to make sure it goes along its way, right out. Okay. Do our valves contract? Do valves contract? No. Because they're not what? What can contract? Muscles. They're not muscles, they're connective tissue. Okay? So muscles are going to just dictate what they do, but they themselves don't have any contractile ability. There's also a big hunk of muscle between the ventricles to keep their blood content separate. And so it's called the interventricular septum. It's between the ventricles. And it also contracts. The base of the heart is this kind of described as flat portion. And the end is called the what? The apex. Right. What questions do you have about this kind of gross anatomy of the heart? All right. We already talked about the job of the valves. They're to make sure the blood goes in one direction, or described here as unidirectional. We'll make sure the blood going in the right direction. I've got a couple of images to help visualize those. We also discussed a moment ago about our valves. Have they passively open and close? They don't do what? They don't contract. The activity of the heart is going to dictate whether they open or close, but they themselves are not contractile. We have a very simple image from a previous textbook, which I've used. We notice when the pressure is greater, on one side, the blood pressure physically opens the door, okay, the valves. Okay. But once that pressure gradient reverses, like we can see in the second panel, the way that the valves are designed, they kind of overlap, and they can't, right, if they're built right, they wouldn't flip back and let the blood go backwards, and so they just keep going in one direction. We're going to learn in the 4th unit that our valves are not only in our heart. We'll see some in some veins as well. So they're in multiple locations. It's a really cool image of some engineered in the lab, some valves. This is the aortic valve. You can see on the far left the valve is open. You notice as it begins to close, and then in the far right, the closed state. So it's actually just kind of sealed, if you will. We've already talked about some primary characteristics of cardiac tissue as well. The cardiac tissue we know is striated. It's short, okay, like our smooth muscle tissue, so it's not like you don't have one cardiac tissue that runs the length of the entire heart. The cells are tiny. And I have the word fat, but I'd like you to change that word with the word broad. They're broad. They're not, not fatty. I meant broad. They're wide. They branch as well. So they're not just parallel like we see in skeletal muscle. They're attached and they turn left, they turn right. Kind of like, to give you a visual, have you ever taken like a towel, like a dish towel? You know how you could get it wet, or even if it's dry or whatever, you could twist it. So the cells of our heart are kind of not in a sheet, but they're kind of more twisted in the heart, if you will. They still work, but they're just branched is what I'm trying to illustrate. They're not parallel with one another. And they are interconnected. And what I'm describing there is the role of the gap junctions. Okay? So we've got a functional syncytium. Cardiac tissue is also multinucleated, so these cells cannot. What can our cells do? Because they have two nuclei, what can this? They can divide. And so if you lose some heart tissue, it's done, right? If you have a heart attack and you lose that without any intervention, and that's why it's one reason why it's important to try to avoid heart attack. The interconnection is between individual cells is an area called the intercalated disc, which you identified in your handout that you turned in yesterday. And it anchors cells through the use of desmosomes and then also gap junctions. So the desmosomes and the gap junctions are located at these regions called the intercalated disc. In your guided notes, you'll see an image, and it's trying to illustrate the branchingness, the shortness, interconnected property of your cardiac tissue. These cells have been what's called teased. So samples have been made and they've been teased apart. Like if you've ever done your hair right, teased, separated, right? So these cells have been separated so that we can see them a little better instead of all jammed up on one another. A couple of things we can see in this image, the striations. You can also see the intercalated disc, these darker staining pink regions where the gap junctions and the desmosomes are in high concentration. You can also see how short the tissue is. It's not real long. And then can you kind of see the branching, how the cells are at angles, if you will. If we zoom in, like kind of like a cartoonized image of a cell, we look at the intercalated disc. Look at how the cells adjoin one another. What's going on there? What kind of structure do we have there? At the intercalated disc. Inter means between, right? And notice how it's not a flat surface abutted. And we've got a big surface area. You can kind of see it folding, if you will. The two cell membranes kind of are adhered and connected well. Because these cells are always generating a lot of tension and a lot of force, and we don't want them to do what? We don't want them to tear. We like our heart untorn. And so the protein, right, or the junctions there at the heart try to prevent that. If we look at the metabolism of the heart, it prefers aerobic respiration. It does have a backup mechanism, but it prefers aerobic respiration. It doesn't prefer fermentation. We also identified that the cells do not divide after a period of development, and so our cells cannot undergo what phase? Cardiac cells cannot undergo, what's another way to say that? They cannot undergo hyperplasia, good job. But they can undergo what? Hypertrophy, okay? Which makes sense. Have you ever heard someone who has an enlarged heart? You ever known someone with that condition? The cells just get larger in that scenario, but they didn't increase cell numbers of the heart. The bulk of the heart is contractile cells, or contractile cardiac tissue, and then a small amount are our pacemaker cells. Here it says autorhythmic. Your book uses the term autorhythmic. We've used the word before, pacemaker. You may also use the term nodes. What nodes are our pacemaker cells? Something called the SA nodes. Anybody know the other one? AV. So I could say autorhythmic cells, I could say pacemaker cells, I could say SA node, I could also be talking about the AV node as well. But these are muscle cells. Let's just go ahead and put our lecture out there. These are cells that will spontaneously, they're pacemaker cells, they will pacemaker cells spontaneously, no? Conduct action potentials. The contractile cells are the ones that contract. Okay. So we can see that it's certainly intrinsically regulated, but we know by definite that the autonomic nervous system dually innervates the heart. I can't remember if we're talking about it here or in Unit 4. Yeah. Tomorrow we'll talk about how the autonomic nervous system regulates the heart as well. Because our pacemaker cells are there and all our cells are connected by gap junctions, we can call the heart also a functional syncytium, which we learned, I used a moment ago about smooth muscle. It works as a, I could say functional syncytium, or what word to be used? A single unit. I can call it a single unit, I can call it the heart, I can call it a functional syncytium, but it's this giant organ working in a coordinated fashion because all the cells are physically interconnected with one another. You okay on the kind of basic properties of the heart? We'll look at some details. The way that the heart works intrinsically is by use of these pacemaker cells. It's been fairly well characterized, so we're going to actually look at the electrical events of the pacemaker cells. We've already stated that because they're pacemakers, they spontaneously depolarize or generate what's called pacemaker potentials. It's just a little bit more specific. It's potential changes in our pacemaker cells. So these cells continually depolarize, and they initiate measurable action potentials. Here's the deal, the confusing bit, potentially confusing bit if you don't take my heat of caution, about the heart. Up till now, depolarization always is influx of what? Always sodium. Not the case here in the pacemaker cells. So specifically, the SA node, specifically the AV node, those cells are leaky to, what do you see up there? Calcium. Make a huge star so you don't get this confused. Calcium leaks into our SA node, leaks into our AV node, causing it to depolarize and cross threshold. Cross threshold, generate an action potential, go back to rest, cross threshold. It just continuously. Hence the name pacemaker cell. Yes, because they are pacemakers. Okay. So I think your book calls it autorhythmic cells, but it's our pacemaker cells. Those are the SA node those are the AV node. Very good. Additional clarification? So the cells of the heart that set the pace of the heart depolarize, but not to our familiar ion. They depolarize because they're leaky to calcium. Very good. You have a similar image in your handout. In your guided notes, I want you to clearly label there on page 2 of your guided notes, what type of cells is this in? Pacemaker cells of the heart. Label that so you don't get it confused. This is very much different than anything we've seen so far. Pacemaker cells, you'll notice, depolarize beyond threshold due to our influx of calcium. The upward swing is due to calcium. And then we cross it, and then we let more calcium into the cell. Depolarization, and then the familiar bit, repolarization, hyperpolarization, and then back up to crossing threshold again. More calcium in. Are you okay on a pacemaker cell? That 1% of the heart, they're permeable to calcium and generate action potentials. The bulk of the heart, though, is not pacemaker cells, but what type of cells? Contractile cells. So the muscle of the atria, the muscle of the ventricles, the muscle of the interventricular septum, they will also depolarize. In order to excite them, though, we have some other cells. Zip over that. Excuse me, I'm getting ahead of myself. I've got some real images of the area of the heart that is considered to be the pacemaker. On the far left is just a zoom in of the area on the right side of the heart where the atria is. Right atrium. And you can't, there's not like a big red flag like pacemaker here. Okay, there's no way to really tell the difference, or at least I don't know how to tell where the pacemaker would be, because it's part of the heart. Okay? Inside of the cell, they are built differently, but superficially, I can't tell. Okay, but right where the vena cava dumps into the right atrium is where the pacemaker cells are located. Okay? If we look at a histological view of the heart, then we can see some intracellular differences. This is right over here, right where the pacemaker is located. If you look on the far right, you can see the differences that you can see staining, more red, like more proteins, where it's less densely packed, right, less proteins. And if you look really closely, kind of intermediate in staining, you can see what's now been pointed out for us, the area that's called the SA node. Okay, so it's a really minute difference of the cells that make up the pacemaker, but they are physically connected to all of our contractile cells. Some of this will be a little bit repetitive because we started incorporating those handouts and kind of warmed up to it, if you will. But the cells of the SA node are physically connected to the contractile cells through what's called conduction fibers. They also don't contract like our pacemakers, but they don't generate the action potential, they just conduct the action potential. So we have these cells that are muscle cells, but they're more like a what? Kind of like a what? They don't contract, they conduct action potentials, they're kind of like what? Kind of like nerves. But they're muscle cells. But they physically connect the areas of that right where that SA node is to the rest of the heart. You can call them conducting fibers. From lab, the ends of them you called, from your pre-lab today, the Purkinje fibers. Okay. Those are the cells that physically conduct action potentials from one spot to the rest of the heart. They don't contract, but they do conduct action potentials that were set by our pacemaker or autorhythmic cells. If we look at some images of the heart, we can see some staining differences. And the areas that are stained a little bit lighter are the areas where the conducting fibers are located. And we have outer musculature and intermuscular. Okay, so we have these conducting fibers, or we'll call Purkinje fibers, that connect the myocardium, the muscle of the heart, and is another musculature layer, but they are physically connected by gap junctions. Does the role of the conducting fibers make some sense, or do I need to present it differently? Thumbs up, thumbs down? Doing okay? Figure 1310 tries to help illustrate where these conducting fibers are located. In this image, what color are they? The yellow things. Okay. The pacemakers are also yellow. Okay. So we have an SA node and our AV node. But the conducting fibers are highlighted in yellow. They again would not be distinct. So like when we do our heart dissection today or tomorrow, they're going, oh, there's a Purkinje fiber. You can't see it. It looks like the muscle itself because it's cardiac tissue. It just doesn't have the thin and thick filaments. It's permeable to cations. But it's a way to physically connect point A to point B. This image is okay, but it'd be nice if they showed the conducting fibers. There's also some that extend between the atria. So they're all in all chambers of the heart. I have a big star here because this is going to tie into lab today or tomorrow. So generally, we're hitting it right about now, so the pace is kind of off a little bit. So if you've done your pre-lab for today, the pace of the heart is started at which structure? Which one is like the pacemaker? The SA node. Conducts then to the AV node. SA conducts to the AV, the atrioventricular node. Those conducting fibers then do what? Branch to the left and to the right. And then they continue, and the end things are called Purkinje fibers. So you might want to put a star there as well. Reviewing lab number, what is that, 9? That we're doing this week today. What we're going to do in lab today or tomorrow is pick up the result of those electrical action potentials at the surface of the body using a device called electrocardiogram. You can call it EKG, K for the way they do it in Europe, or E. Sometimes I think people over here are starting to convert it to ECG. Does anybody work in the hospital? K or G? Kers. Okay, so still using EKG. So some people use ECG for the K or the C, cardio. And we're picking up the electrical activity as a result of the internal electrical events of the heart. But if you work there, are you probing the heart?
No, for the EKG?
Correct.
No.
No. Okay. So when we do our EKG interpretation, are we measuring action potentials directly? Mm-hmm. No. We're measuring the result of the action potentials. It's just, you can pick it up throughout the body at the surface of the body. And so we're going to have 2 leads, technically 3. How many do you use in your setting?
8.
8?
Yeah, 8.
8, and you can make upwards of like 12, okay? The more leads you have, electrical contacts, the more what do you think your data is? Accurate. So our data is probably not going to be very accurate today. So don't panic if your data doesn't, I can almost guarantee your data is not going to look textbook. So don't overthink it, okay? The time is going to be accurate, but not the way that it looks. So don't overanalyze it, okay? And so the heart continually beats, and so it really just depends on when you put it on when you'll start reading the EKG. And we know that the depolarization event starts where, you said, at the SA node. And so as the SA node begins to depolarize, the muscle cells which it's connected to also begin to depolarize because they're all connected by gap junctions. Okay. And this depolarization event can be measured first with the EKG. So the first wave that we'll see reading an EKG, and why don't y'all go ahead and add notes so we don't have to do it in lab. Okay. So these things are called waves. Up means a positive deflection, down means a negative deflection, right? Positive versus a negative lead. And so we're going to have these different waves that we're going to interpret. The first wave is called the what letter? P wave. So label this the P wave. It's kind of highlighted for us. And if it's too squished, if you need to write on another piece of paper, that might be useful too. During the P wave, which structures are beginning to depolarize? Which part of the heart? The atria. Starts over the right atria, but because our conducting fibers, which atria will also? The left. And so full depolarization of our atria completes what's called the what? P wave. Nomenclature P doesn't correspond with some letter like potential or whatever, right? Some word. It's just in sequence, like when you take algebra, you do A, B, C, X, Y, Z, whatever. This is going to go P through what? T. Okay, just in sequence. So the P wave represents atria doing what? Depolarizing. If these cells depolarize, the chambers will do what? They're going to contract. They do contract. Does anybody remember the word we could use to summarize the contraction and the emptying of the blood? Systole. Very good. So during the P wave, that represents the electrical depolarization of the atria, but also tells us that they are entering into systole. So during the P wave, the atria are depolarizing and are in systole. Notice the AV node is now beginning to do what? It is beginning to depolarize as indicated or illustrated by the yellow. Okay, the yellow is going to represent our depolarization. Before we excite the ventricles, though, what do you think we should do? We're just atria and systole. Before we put the ventricles in systole, atria should probably go into diastole. Okay, and so the atria should enter into diastole, which we can kind of see here as the chambers are beginning to go back to the kind of non-colored area. As the atria begin to repolarize and enter into, y'all said, diastole, notice that the conducting fibers that extend down to the ventricles are now doing what? They're depolarizing. But we have so much area to cover, ventricles are not fully in what? We're just exciting them. They're not fully into what phase? They're not fully into, did someone say contraction? Yeah, they're not in systole yet. Atria just finished up systole, they're beginning diastole. The ventricles were in diastole, but now they're about to go into systole. Okay. And so this happens during what's called the what? First, it is part of the complex, but first we get into the P, then Q wave. Not much later, we do get full repolarization of the heart, or the atria, excuse me. The atria completely repolarize and enter into diastole. And then very soon, electrically, the ventricles will depolarize and enter into systole. These events are nearly simultaneous and do take place during what's called the QRS complex. QRS complex. So a lot's happening during the QRS complex. Even though it doesn't take long, it's happening. There's a lot of events happening in that short amount of time. During the QRS complex, the atria repolarize and enter into, you said, diastole. Yeah, the ventricles. Did I say depolarize? No. Okay. So the atria repolarize, make sure if I didn't misspeak. The atria repolarize while the ventricles are depolarizing and physically contracting, sending blood out to either the systemic circuit or to the pulmonary circuit at the same time. What do you think has to happen next? What do we have to do? Just relax the atria and just contracted the ventricles. The ventricles better go back to relax. Okay. And so there's not an extra image there, but during the what wave? The T wave, the ventricles repolarize and enter into diastole. We just walked through the major phases or events of an EKG under normal conditions, the P wave, QRS complex, and the T wave. But we'll elaborate more when we look at the cardiac cycle. It's basically first the atria contract, and then they relax, so that then the ventricles can contract and then relax. Okay? So it allows for the heart to act as a what? Pump. Before I move on, what questions do you have about the EKG, what the phases represent? There will be a little bit of a lag or a latent phase, and then what happens? Starts over. Every beat. This little animation illustrates the depolarization events and repolarization. You can see the red representing where the series of excitation is. You could also see the status change of the atria versus the ventricles in the contracted versus relaxed state. And then the corresponding EKG below. So first atria depolarized during the P wave, passed on to the AV node, interventricular septum, those conducting fibers. And you notice the contraction operates bottom up of the ventricle, propelling the blood out. In this case, we can see out into the systemic circuit. Okay. So atria P wave, SA node, Q, ventricles RS, okay. Ventricles relax, T wave, repolarization. Also notice what happens at the valves when the atria contract. The AV valves are what? They're what? When the atria contract, the AV valves are open. And then when the ventricles contract, the AV valves are closed. Does that help a little bit? Bring about any questions? Okay. So that's just a generic looking textbook kind of EKG pattern, which we'll measure in lab today or tomorrow, whichever lab you're in. Our pacemaker cells, we have already identified as our SA and our AV node. Without any extrinsic stimulus, the SA node depolarizes at about 70 to 80 action potentials per minute, which indicates what? Kind of correlate to beats almost. That's your what? Kind of like your heart rate. But your heart rate's probably a little bit slower than that, so which nervous system can override this? Your heart rate could be a little bit slower than that. Which nervous system can control that? Parasympathetic. Slows the heart. Very good. You'll notice that the rate is slowed down a little bit at the AV node as well. Our conducting fibers, the bundle of His and the Purkinje fibers, also slow down the rate of conduction. So the pacemaker has the fastest rate of depolarization and therefore sets the what for the heart? Sets the what? The pace, as the name implies. We already know this is repetitive. The cells are linked by gap junctions. And I just mentioned that those cells that depolarize the fastest are the ones that are control the electrical and therefore contractile activity of the rest of the heart. Okay. All right, which is restated there. You okay to move on? We look at the relationship between those pacemaker cells and those contractile cells. I know that you became introduced to this in handout 5 that you've already submitted. You know that the pacemaker is connected by gap junctions and it's repetitive because it's kind of important, right? How we can see the electrical activity flowing between cells of the heart. But if we look at, if we step away from the pacemaker cells and look at the contractile cells, they also generate action potentials. That's result of what's going on. And they are, the cardiac muscle cells have what's considered the contractile cells, have a long refractory period. We've used that term before. We didn't mention it in our skeletal muscle, but it's important to acknowledge here in the cardiac muscle. It has about a quarter of a second refractory period. What it prevents are these types of contractions called tetanic contractions, or tetany. Not the disease, but does anybody know what tetanus means? They must have never had what do you think the word think about the disease, the symptoms of the disease if you have it locked up, seized up, right? Would it be a good idea if we could lock up the cardiac muscle? No. Tetanic contractions are okay in, say, skeletal muscle. So if I asked you to hold, you're in an action movie, and you've got a grenade in your hand, and the pin's been pulled, so you need to hold the little thing, right? Is it okay to hold the contractions for a long time? You're going to hold the pin so it doesn't let go. Yeah, hold it, right? You can hold it. Let's hold it till someone comes helps you, right? You can hold that, okay? And you're not going to spontaneously, without the help of the thing, die, right? Because your skeletal muscles are contracted, right? So you can sustain a contraction for a long period of time with skeletal muscles and be okay. Conversely, though, in the heart, what if your heart muscles sustain a contraction? It stopped working. It's no longer acting as a pump. You're not pumping blood. You're going to very soon die. We don't want to have that happen accidentally in the heart, so the heart has this kind of protective mechanism, and it's these things called a refractory period. Okay. And we'll address how that happens today, and if we need to better detail it tomorrow, we will. But the physical mechanics of contraction are similar to that in skeletal muscle. What I mean there is the thin and thick filaments, calcium, right, type of, right, calcium binds to troponin, expose the binding sites, ATP powers, right, the myosin cross-bridge is what that's in reference to. But if we look at the role of that refractory period. Okay? The contractile cells, they'll depolarize similarly from about minus 90 millivolts up to about 30 millivolts. They'll depolarize due to our familiar ion, what? Sodium. Okay, so this is different than the pacemaker cells. They'll depolarize due to the influx of sodium. But instead of immediately repolarizing, going back to rest, the cells stay excited. The contractile cells stay excited or depolarized for a period of time. And it's due to the influx of calcium. So we've got two things going on here. The cell depolarizes, generates an action potential, sodium goes in, we're familiar with that. But then gated channels open so that we let calcium in. Calcium has what charge? Positive. And so now we just keep it excited. Okay. Eventually the gated channels will close, and then what? Then the heart can, the muscles can repolarize. Okay. And then go back to resting membrane potential. Okay. But what's unique to us is that what's described as plateau phase, okay, where we're maintaining this excited depolarized state for a period of time. If we look at it, I need to flip back to that image. Sorry about the placement. If we look at it in a plot, figure 13 helps illustrate the electrical events of your contractile cell. So on the image on page 3 of your guided notes, label this. We're clearly differentiating this. These are what type of cells? This action potential is really characteristic. I see this, I see what type of cell? Contractile cells of what organ are we in? Heart. Okay. So cardiac contractile cells. What other word can we use based on lab? We can call these contractile cells. You want to pick up any other alternative name for the muscle of the heart? Myocardiac. Okay, myocardial. The cells of the cells that are what myo means, muscle. The muscle, not the pacemakers. Or I could say the cells of the atria. I'm going to say the cells of the ventricle. The bulk of the heart. Those cells, when the action potential floods through them, sodium floods in initially, and then they let in calcium. To prevent what? Not to prevent it, but to do what with it? Because eventually it's going to repolarize. Just to slow it down, delay it. Why do we want to delay repolarization? To prevent tetany. So make sure the heart, that muscle contracts, and make sure it does what? Goes into systole, then make sure it goes into diastole. That plateau phase ensures, it makes sure that after contraction the muscle is going to relax. Without it, we could sustain contractions like our skeletal muscles or smooth muscles or whatever, which we do not want to happen in the heart. I'm going to conclude our discussion here. I do encourage, or will encourage you to get into Chapter 6, although it's not due. Do the questions correlating with Chapter 13 for homework number 6. And then any questions you have from that, bring those for class tomorrow. And tomorrow we'll conclude Chapter 13, and on Monday, good old exam.